de Frontiers del año.
Recording in
Y realmente es un
progress.
placer presentarles a la doctora Alicia Martin, que nos acompaña hoy de la Universidad Harvard. La doctora Martin estudió bioingeniería en la Universidad de Washington para su licenciatura y después su maestría la hizo en biomedicina, en bioinformática en medicina en Stanford, su doctorado en genética también en Stanford y después se movió a Boston, a la Hospital General de Massachusetts, donde realizó su primera estancia postdoctoral, que concluyó en el 2018 y a partir de ahí, del 2018 al 2021, se desempeñó como instructor en investigación en el Hospital General de Massachusetts, también como instructor de medicina en la Escuela de Medicina de Harvard. En el 2022 se convierte ya en investigador asistente en el Hospital General de Massachusetts. En el mismo año es también profesor asistente de la Escuela de Medicina de Harvard y miembro del Broad Institute del MIT. La doctora Martin ha recibido muchos premios por su trabajo en entender la genómica y la genética humana y los polimorfismos asociados a enfermedades, y solamente voy a leer un par de ellos. En el 2022 recibió el SCLAR Award para la innovación y la integración de la genética a las enfermedades psiquiátricas. Y en el 2023 recibió el reconocimiento a la Early Career Investigator en Genetics and Genomics. La investigación de la doctora Martín se centra en abordar los problemas de Gracias. Se centra, como decía, en integrar la información generada por los ensayos de GWAS para que sean aplicables a todas las poblaciones y, de hecho, sus estudios principalmente es promover la equidad en la investigación genómica global, lidera y colabora estudios a gran escala como el proyecto del Pan UK Biobank y la iniciativa global de metaanálisis de biobancos, que incluyen datos de muchísimas secuenciaciones y también datos genéticos en estudios como NeuroGap en África y PUMAS en poblaciones subrepresentadas en estudios de enfermedades mentales. Además de generar recursos comunitarios, como Genome AD, contribuye a la capacitación en genética y estadística y bioinformática en el sur de África y Asia. Tiene muchos proyectos realmente donde impulsa la genómica en países donde no se desarrollan estas ramas, sobre todo tratando de entender cómo la variabilidad genética de esas poblaciones está subrepresentada en los estudios genómicos que generalmente son dominados por las ancestrías europeas y que por lo tanto esos datos no se pueden extrapolar a las diferentes poblaciones. La doctora, a través de liderar estos grupos y estos consorcios de investigación, ha publicado muchísimos artículos en revistas muy renombradas, sin descuidar también la formación de recursos humanos. It's a real pleasure, and I thank you very much for your accepting the invitation and me here today.
Thank you.
Thank you.
Okay, well, thank you so much for that wonderful introduction that I understood a little bit of, but as a non-Spanish speaker, or at least having only taken a year or two of Spanish, Thank you so much for helping translate the type of work that we do to everybody, everybody here. And thank you so much for the warm welcome, especially to the students who I had a real nice privilege talking with yesterday. Okay, so I'm going to be talking about a breadth of genetic studies. So the first part of this, we're going to be talking about kind of an introduction linking human evolution to complex trait genetics. Then we're going to dive into biobank scale studies across traits and ancestries to empower genetic architecture studies and identify new biological discoveries. Then we'll be kind of shifting gears a little bit to the brain, where because I'm based at the Broad Institute, where we focus particularly on psychiatric disorders in the Stanley Center, we'll be talking about diversifying psychiatric genomic studies to empower insights into particularly severe mental illnesses like schizophrenia and bipolar disorder to empower evolutionary studies as well. And then we'll be ending the science portion, the new research on dissecting pleiotropy, the relationships between multiple different traits and the genetics that underlie shared sort of etiologies of multiple different diseases, and delineating genetics from environmental risk factors, particularly for respiratory disease, to understand why there are such massive disparities in lung diseases across populations. Is it genetics? Is there some environmental component? How are these kind of shaking out? Then we'll end with some key takeaways and conclusion. So I did my training in human evolutionary genetics. And so as a geneticist thinking through an evolutionary lens, I like to start at the beginning. And the beginning for our species means we need to start in Africa, where our species originated. So on the order of hundreds of thousands of years ago, we originated in Africa, took a subset of genetic variation with us as we moved into the Middle East, into Eurasia, and subsequently into the Americas. So this population history over the course of the past 100,000 plus years has shaped our genetic variation and is really important then to consider when we're designing our genetic studies. For example, because of this history, the average out of Africa population has about 1 million fewer genetic variants than the average African population. Notably, there's more variation within than between than between populations. And this human history over the past 100,000 or so years informs this landscape of genetic variation across the world and is one of the key ingredients when we're designing our genetic studies. So it's really important to take into consideration for how we're performing these studies. Over the past decade or two, we've really had an unbelievable pace and explosion of progress in human genetics. Starting from around 2000, when we completed the first sequence of the Human Genome Project, which cost over $3 billion, we're now sequencing routinely genomes for about $300. So that decrease in cost has made tremendous impacts on the scale of studies that we're able to perform and has led to amazing revolutions. And so I can't think of a more exciting time to be doing science just based on the infrastructure and resources that we have in human genetics. I don't know that we would have had mRNA vaccines or CRISPR technologies or so many fantastic and exciting opportunities without this type of progress. And I see this as really fueling two missions in genetics. The first is providing biological hypotheses from, very importantly, unbiased associations in genetics. So we're not going after our targets or our pet hypotheses. We're scanning the entire genome to learn about what is the most important contributor to these particular diseases or pathways. So I see this as informing the basis for downstream functional analysis and even offering potential therapeutic insights, as we've learned from drug discovery studies that those therapeutics that are supported by genetic evidence have fewer side effects and are more likely to be approved. But a huge problem in the field is the vast Eurocentricity of these data sets. So currently about 80% plus of participants in large-scale genetic studies are of European descent. This is far out of step with the global population, where about 16% of the world's population has primarily European ancestry. This hasn't changed much. So we highlighted this problem around 2019, but this had been highlighted also a decade earlier. So calls for diversifying progress have kind of fallen quietly. Notably, though, over this whole time span, we haven't seen diversifying, an increase in diversity in our genetic studies, even though they've exploded in sample size. So this is a really core and fundamental problem because, as I mentioned on that map, if we have less genetic variation from doing sort of biased ascertainment of our genetic studies, we're then destined to find fewer genetic associations that are going to be less generalizable across populations. So the core question that my lab is really interested in and many other folks are working on is how do ancestry study biases in genetics impact the generalizability of knowledge across populations? Now, with this kind of frame in mind, I want to go over a couple of important considerations. So the first is that causal variant effects are largely shared across populations. So the reason somebody has a heart attack in one population is going to be the same reason somebody has a heart attack in another population. We shouldn't be surprised that the genetic risk factors that are contributing to risk of heart attack are also shared in different populations, though they may be different in frequencies. So coming back to that human map of human history, things like allele frequencies across populations differ. Things like the correlation structure of genetic variants in the genome differ as a consequence of our human history. There are also other complicated differences like our environments and natural selection and other complex factors that we have not a very good grasp on the field right now, how they're influencing complex traits across populations. So with this question in mind, one of the tools that we've been using to try to understand the generalizability of genetic technologies is called a polygenic score. So a polygenic score effectively takes GWAS that have been conducted previously or genome-wide association studies. They take the effect sizes that have been estimated in those. They multiply those by genotypes in a new cohort. They add them up across the genome and they predict some traits. So when we've done something like this using data from the UK Biobank, a relatively homogeneous cohort, and we use European ancestry data, so the bulk of the data, to predict traits across different populations, what we see is that we're predicting different traits far more accurately in European ancestry populations than elsewhere. So for example, we're predicting these traits, a whole variety of traits, dozens here of mostly blood panel and anthropometric traits, about twice as accurately in European ancestry populations as in East Asian ancestry populations, and about 4 to 5 times as accurately as in African ancestry populations. This is simply a function of genetic divergence between populations and reflects our human history. This is really important. This is an exciting biomarker. I can't think of any type of biomarker that's as informative across so many different types of diseases with just a DNA test or a GWAS array that's been run in one individual. You can predict dozens of different traits based on all of the GWAS that have been done before. But I see this as the single biggest challenge to implementation of genetic technologies in precision medicine, because we're simply less able to predict traits accurately across populations, not because of any consequence of their health risks, but simply due to their ancestors. So this is a particularly pernicious problem across many areas of biomedicine, but is especially challenging in genetics because of the profound effect of ancestry on our DNA. Okay, so there's some really big challenges and opportunities that I think we're going to be able to sort of address in the coming years. So genetics is moving at warp speed, it's moving incredibly quickly, and so we've got some amazing opportunities if we diversify our genetic studies. The first of these big opportunities is identifying new biology through novel genetic variants that are present in underrepresented populations in genetics, but not in the largest and best studied populations. We've already seen examples of these playing out into therapeutics. So for example, PCSK9 inhibitors are sort of the poster child of the first genetic success in bringing a drug to market that we would not have identified otherwise. This study was conducted in about 5,000 African Americans that identified this loss of function variant in PCSK9 that led to LDL cholesterol lowering medications that work even when statins don't if people are not responding well. And that's really exciting because we needed sample sizes on the order of hundreds of thousands of individuals to identify this gene well in European ancestry populations. So we were able to start this sort of whole therapeutic pipeline process because of where this study was done and because of the variants that were present in this unique population. We're also better able to pinpoint causal variants because of the differences in correlation structure across the genome. So I mentioned that causal variants are largely shared, but correlations across variants are going to differ. So we can leverage that difference in correlation to understand whether there's heterogeneity across populations to determine, is this gene likely causal? Is this variant likely causal or not? If you see a lot of heterogeneity across populations, that's probably not the variant you're looking for, and that's probably not the causal variant. There's a low degree of heterogeneity for a particular genetic variant across populations. Maybe that has more likely chance of being a causal variant. So we can leverage these differences to also inform which genetic variants we should be following up on in functional assays to try to tease apart what its mechanism is and how likely that is to be causal for some downstream disease of interest. We can also bring together genetic and environmental determinants of health. So without understanding the breadth of genetics, we're kind of lost, but also without understanding the breadth of environmental variation, we're a little bit lost. And then lastly, genetic risk prediction accuracy requires some reasonable sample size to be able to identify and improve genetic risk prediction accuracy for everybody, and especially in underrepresented populations. So to get there, there's a few needs, and apologies for the recap for the students. Things will get newer in a moment. One of the first things that we need are statistical methods that do not assume the same degree of homogeneity as we have traditionally in the past. So a lot of our statistical methods require that you fall within some rough degree of variation in a population, which means we need to do studies within a relatively homogeneous group. Moving beyond that requires newer and more advanced statistical methods. The next area is in data and community resources. So we need to make better use of the data that we already have and not leave out underrepresented populations from these large studies and biobanks that have already been set up. We also need to collate data across existing boundaries. No single lab is going to solve the genetic basis of one disease. Therefore, we need to work together and break down sort of fiefdoms in genetics. We need to work together and make sure that our data are all being brought together in a harmonized way to advance genetic progress most successfully. The genetic data that we have is also not representative of the field. So the best way forward is to grow to address gaps in our studies. And then we also need to develop some supporting resources to ensure the downstream functional dissection of our studies are equally diverse so that we're, for example, if we're looking for an EQTL that's not present in a population, we have a resource where we can identify those variants and their functional effects. And then lastly, we really need to increase research capacity and the future generation of diverse leaders. So when I showed you that picture of representation in genetic studies, that's not reflective of global diversity at all. So we started there. It's also not reflective of US diversity, which given that the NIH is one of the largest biomedical funders of research in the world, maybe you would think that at least we should start with meeting US-based diversity, but we're nowhere close to that either. We're okay. Do you need me to wait a second? Oh, great. We are, in fact, really closely representing the diversity of NIH-funded investigators. So if we're hoping to change the representation of genetic diversity in these studies, likely we should start with who is conducting those genetic studies and ensure that the representation in the field is diverse as well. Okay, so now we're going to get into some of the science. I put together this presentation. Everything in here is going to be unpublished, but please feel free to talk about it. Everything is basically pre-printed, so we're in like kind of the early stages of getting everything published. We're going to start with biobank scale studies, and these are really empowering, really massive studies of genetic diseases that we've never been able to do at this scale before. This will include things like type 2 diabetes, asthma, cardiovascular disease, all of these sorts of traits where we can use electronic health records that have been collected across different resources and try to understand their genetic basis. So one of the broadest and most widely available and best used data sets is the UK Biobank project. The UK Biobank is a very homogeneous cohort overall, but it's also a study of 500,000 people. And so only about 5% have primarily non-European ancestry, but 5% of 500,000 people is still rather large numbers. So we still have over 20,000 participants in the UK Biobank who have primarily non-European ancestry. So that means we're leaving a lot on the table if we don't analyze those participants. So we use the UK Biobank data to make better use of the underrepresented populations in this group. We ran over 16,000 GWAS of over 7,000 phenotypes with 6 different ancestry groups. And a lot of things that popped out of here are largely shared across populations, as I mentioned. But if we kind of look through, we are getting, if we kind of like, look at how many novel variants we have overall about 71,000 genome-wide significant associations. We have about 34,000 new things that we don't see based on a shared sort of phenotypic category or phenotypic term. If we're a little bit more specific about our phenotype definitions, we see about 5,000 new associations in this study. And then if we start to look at how many are new based on primarily non-European ancestry enrichments, we do see that there are about 3,000 individuals that are novel, including in previous UK Biobank, and we're seeing a couple hundred that would not be found if we hadn't analyzed those 20,000 or so underrepresented ancestry individuals. And kind of looking at how common these are in European ancestries and through this combined meta-analysis across ancestry groups, you would generally expect that things should kind of like fall along this diagonal here. We should mostly see that we're not adding much significance if we're adding 20,000 individuals to a study of 400,000 plus individuals that are of European ancestry. But in fact, we do see a bunch of variants up here that seem to only be identified because they're present in these 20,000 or so individuals. Just kind of a proof of principle that doing these genetic studies in these underrepresented ancestry groups, even if it's far, far smaller than the largest data set, is in fact identifying potentially new biology. So the UK Biobank is not the only data set that's out there, and thankfully there are far more diverse data sets. So one of those that has come up fairly recently is the All of Us research project, or All of Us research program. This is far more diverse, so it's about half European ancestry, but largely also non-European ancestries. Within our lab, we wanted to try to understand whether we're able to increase polygenic score accuracy by combining data from the UK Biobank as well as from All of Us within multi-ancestry studies. So we ran GWAS for a few dozen traits. We harmonized binary endpoints. So these are disease traits. We analyzed a couple or 18 of those and also 14 different quantitative traits. These are things like height, BMI, blood pressure, as well as blood cell traits like white blood cell count, for example. We did GWAS across all of these, and we were really curious about the breadth of traits that we were analyzing. So is there a difference in how well we're going to be able to improve polygenic score accuracy? For example, depending on the ancestry composition that's making up our genetic studies, that was our key original thing that we were interested in. But we were also interested in whether the trait architecture has a big impact. For some traits like height and schizophrenia, for example, it seems like the entire genome is effectively contributing to risk of these or likelihood of these traits. Whereas for other traits like, you know, on the extreme end, a Mendelian disease like cystic fibrosis or sickle cell anemia, you would expect only one gene to be identified here. Some of the traits that we weren't looking at any Mendelian traits, but we're looking at some traits that also have sparser genetic architectures like LDL cholesterol, which are a little bit different from, say, height and schizophrenia. So we conducted a single ancestry GWAS within UK Biobank. We used African ancestry individuals and European ancestry individuals, as well as in all of us. We have admixed American, Hispanic, and Latino populations, African ancestry individuals, European ancestry individuals as well. And then we combine data across UK Biobank and all of us through meta-analysis of these multi-ancestry statistics. Then we calculated polygenic scores and evaluated within a holdout set in all of us how accurately we're predicting these traits. We also wanted to look at individual accuracy across sort of a diverse ancestry spectrum at the individual level. If we're hoping to translate polygenic scores and return any of these results someday in the clinic, we need to be able to take somebody who's got ancestry from 4 different continents and be able to say, here's your polygenic score, and this is what it means for you specifically. So with all of that in mind, this is just showing you across dozens of quantitative traits how accurately you're predicting depending on what our study design was. So we asked how accurately we're going to predict in European ancestry in blue, African ancestry in purple, and admixed American ancestry or Hispanic and Latino ancestry in red. The different traits are shown on the bottom in the same order. The top part is showing you if the discovery GWAS, so where you're training your polygenic score, is coming only from all of us, say an African ancestry, admixed American ancestry, European ancestry, or the combined meta-analysis that brings all of the data together in all of us. Similarly, what happens if you only use UK Biobank data versus what happens if you use all of the data all together? So you might expect that the largest sample size should perform best. In that case, you would expect this row to be lit up in all of these. You would expect boxes around all of these. In fact, what you see is that the European ancestry data tends to do best when you analyze all of the data in UK Biobank only. In African ancestry populations, we tend to see that we're doing a mix. So we're doing well for some of these traits when we're only looking at multi-ancestry all of us data, or when we're combining both data sets. And then also it's a little bit mixed for admixed American populations, although that's sort of a difference between whether you should combine all of us data or whether you should only look at multi-ancestry UK Biobank data. So what does this mean? One thing that we've kind of learned through this process is that heritability actually differs between UK Biobank and all of us. Now, I should note, heritability is not a fixed entity. You might ask, what is the heritability of height? And I would have to ask you, in which population, in which study, et cetera. So if I'm measuring height in inches, I have a slightly less precise measure than if I'm measuring height in centimeters, so I have less phenotypic noise. That will impact my heritability estimates. The more precise our estimates are and the cleaner our estimates are, the higher the heritability should be. The US healthcare system is notoriously messy. We know that there's a mix of inches and centimeters for height, for example, and there's all sorts of differences in how these two cohorts were ascertained. UK Biobank is a volunteer-based cohort. People have done studies comparing it to the UK Biobank or to the UK population on the whole, and have found that this cohort is healthier, wealthier, and higher educated on average than compared to the UK. The All of Us research program, by contrast, tends to be more hospital ascertained, although there's a variety of different recruitment strategies. That means that the average All of Us participant tends to be a little bit sicker on average than the typical American. So this also impacts how we need to be thinking about the interpretation of our results across UK Biobank and All of Us, and in fact across all biobanks around the world or across all cohorts. You really need to be considerate of how different ascertainments are affecting your results. Okay, so then there's sort of some interesting findings that we had based on how accurately and generalizably we're able to predict a variety of different traits. So on the top is how accurately we're able to predict BMI. The solid line is showing you if we use European ancestry training data alone, and the dotted line is showing you if we use the multi-ancestry training data set. There's basically no difference between these lines. Pretty much everything is all lined up on top of each other, indicating that it doesn't really matter. BMI, you're kind of doing the best job by just having the largest sample size. And in fact, you can see that if we're comparing how significant results are in European ancestry results compared to the meta-analysis, everything is kind of lining up along this line, indicating that the multi-ancestry data is not giving you substantially more power to identify large ancestry-enriched effects for BMI. That contrasts with a trait like neutrophil count. So on top, you're seeing very little decay and how accurately you're predicting across different populations when you use the multi-ancestry training data set. If you use the European ancestry training data set, though, things decline rapidly, especially in African ancestry populations. So there it's really important to be considerate of our multi-ancestry data and not to just discard to use the largest European ancestry component of the All of Us data set. Now, why is that true for neutrophil count compared to BMI? Well, for neutrophil count, if we look at what we identify in the European ancestry data compared to what we find in All of Us multi-ancestry data, what we see are some very large ancestry enriched effects. So for example, we very, very cleanly and clearly pull out the Duffy null allele. This is a textbook example of impacts on malaria, but also this benign neutropenia trait that's very common in African ancestry populations. So this has really big impacts for the generalizability across populations, but it also means there's a lot of trait specificity in terms of how we need to be designing our studies to optimally predict different traits across different populations. So unfortunately, the takeaway here is it's a little complicated. Okay, we've also been trying to make available in a similar resource as in UK Biobank an all by all GWAS data set. So we have dozens of different quantitative traits. We also have hundreds of different binary traits, lots of also treatment response measures, exacerbations, all sorts of things that have been pulled from the EHR across different participants. So within the All of Us data set, we have a new release, but in this current release, we have about 250,000 individuals who have a complete EHR as well as genetic data that we can sort of map together and try to understand genetic variant risk for a wide variety of different traits. So within that 250,000 individuals, about 50,000 are of African ancestry, about 40,000 plus are of admixed American ancestry, and over 100,000 are of European ancestry. Putting all of this data together, we have the best power to identify genetic variant, genetic associations in European ancestry populations, but it's also much more powerful for multi-ancestry analysis than what we ever had in UK Biobank. So this is a great resource. A really talented computational biologist analyzed a wide variety of different traits using GWAS, Wenhan Liu. This is sort of the breakdown of different types of traits. So are you looking at personal family history, prescription data? Phe codes are basically aggregations of ICD codes in your electronic health records. So these are the diagnostic criteria for a disease that are present in your EHR. We have a lot of physical lab measurements as well as just basic lab measurements that have also have GWAS now available. So one thing you may be interested in that's sort of a unique power from all of us compared to other data sets is that it has had whole genome sequencing done on all participants. That allows you to look at rare loss of function variants within all of us compared to many other cohorts that simply have genotyping data. When you have rare loss of function variants and you understand what it's associated with, that's much more likely to give you a therapeutic target. You know how to knock down that gene in a mouse model or in a cell line or in any functional model, and you can very easily measure its outcome. The vast majority of GWAS results are non-coding, which gives us a much harder time trying to understand what we should do with this in follow-up studies. One thing that we see is that we're increasing our power for gene burden testing by combining all of us data with the UK Biobank data that has been whole genome sequenced as well. So here on the x-axis is the minus log 10 p-value if we're looking at the UK Biobank data alone. And then on the y-axis, we have the results if we combine the UK Biobank results with the all of us results. And one thing that should be clear from this is that our synonymous results are kind of falling all like along the diagonal here. So we're not really seeing any increased burden of risk within synonymous variants. That's our null and that's our expectation. So this is how we're basically like calibrating our model. For missense variants, they're going to be kind of a combination. So missense variants mean there may be some severe effect on the protein, but it may be pretty functionally benign. There we're seeing something kind of in between where we're seeing like a little bit of enrichment in the meta-analysis, but it's a little bit neutral. And then if we combine this data and look at the most severe deleterious kind of set of variants, these loss of function variants that knock out the function of the protein, there when we add together UK Biobank data and all of us data, we're seeing a lot more significance in the meta-analysis, which is giving us the statistical power to go in and identify new genetic variants that are most closely linked to deleterious effects in genes that cause disease. So these are the most important and low-hanging fruit to go after. There's a huge public resource that we've developed as part of this. So we've released GWAS results in the All of Us Researcher Workbench. There's a public browser as well that you can go and sort of mine these genetic associations with a whole wide variety of diseases, including type 2 diabetes, BMI, LDL cholesterol, and many other things that are of great importance for public health. Okay, so that's some of the broad biobank stuff. I'm going to go a little bit more deeply into some work that we're doing in psychiatric genetics in particular. So we're especially interested in understanding the genetic basis of severe mental illnesses like schizophrenia and bipolar disorder. These are incredibly heritable. They have a similar heritability to height. They're highly genetically correlated. Pharmaceutical companies have kind of abandoned a lot of their programs that are based in severe mental illnesses because they've been met with a series of setbacks. It's far harder to study severe mental illnesses than other disease areas because no one's really willing to part with their brain tissue, which limits pharmaceutical studies for obvious reasons, and it would be very unethical to try to move those forward. So genetics provides one of the most promising lines of inquiry for understanding these diseases and trying to move therapies forward. So to achieve larger genetic studies across a breadth of populations, we've launched this Populations Underrepresented in Mental Illness Association Studies, or the PUMA study. So across several African and Latin American countries, including Kenya, Uganda, Ethiopia, and South Africa, as well as Colombia and Brazil, and then a lot of underrepresented populations in the US, including African American, Hispanic, and Latino populations. We have collated together a sample bank of 180,000 participants with cases and controls, about half cases, half controls. And we've also sequenced all of these individuals using a unique blended genome exome strategy that we developed specifically for underrepresented populations in genomics. We're in the process of analyzing our associations between genotype and severe mental illness. And there's some interesting and new findings that are popping up here. For example, within NeuroGap, we're seeing genes that have been nominated in European ancestry populations, but with different genetic variants that are only common in African ancestry populations and seem to be absent everywhere else. These are really interesting potential therapeutic targets to follow up on. So we developed the blended genome exome sequencing strategy instead of GWAS arrays for this big study. We changed our data generation strategy because African ancestry populations, for example, have a lot more genetic variation than other populations. And additionally, GWAS arrays tend to have Eurocentric allele frequencies, so they're more common in European ancestry populations than elsewhere, which means even if you have everything all equal, you're going to have worse accuracy in trying to understand what genetic variants are that you're not measuring on the GWAS array through things like imputation. So this strategy sequences the whole exome at about 30x, and then the whole genome at about 2 to 3x. There's several big advantages to this. The primary one being sort of a Goldilocks approach. So we get rare coding variants that are the easiest to understand, interpret, and go after. We also get unbiased common variant capture, which was the original reason we designed this. And this is also only $100 per sample, which is comparable. It's a little bit more expensive than genotyping arrays, but it's far cheaper than sequencing. So just to tell you like a little bit more about this sequencing strategy, the blended genome exome strategy really is kind of like this Goldilocks approach. We've evaluated how well we're doing at imputing genetic variants in the non-coding region as well as in the coding region. So here I'm comparing a really commonly used genotyping array, which is the Illumina global screening array, but it's not really very global. It mostly has European ancestry allele ascertainment. And then I'm comparing with sort of the gold standard 30x short read whole genome sequencing. So for less common variants, we're not imputing quite as well. That's totally to be expected compared to deep whole genome sequencing, but we're doing quite well compared to a genotyping array. We're imputing almost twice as, or about twice as many variants. For common variants, we're imputing almost all of the common variants that you would see in whole genome sequencing and are doing quite a bit better than the GWAS arrays as well. What's really interesting though is we're also getting almost complete capture of rare coding variants, which these are the most important and interpretable to be able to go after to understand disease biology. And we're also capturing these common coding variants much better than we would with a genotyping array. A GWAS array like the Illumina GSA is about 20% of the cost of a deep whole genome sequence. And then the BGE that gets you a lot of these like unique scientific advantages is about 30%. So it's a little bit more, but gives you a lot of these advantages as well. Okay, so we've applied this approach and we're looking at the association between genetic variants and schizophrenia and bipolar and psychotic risk, particularly in this study called the NeuroGap psychosis. This is the part of the study that I've been most integrally involved in. NeuroGap is short for the Neuropsychiatric Genetics in African Populations. This is a major global initiative launched by the Stanley Center for Psychiatric Research around 2015. So it's now been about 10 years since we very first launched this study, or basically filled out our IRB forms. It took a couple years to get approval from that. COVID slowed a lot of things down. But I just want to mention that we have over 43,000 participants that have been recruited and sequenced as part of this study, which is far faster than we would have expected to be able to hit these large goals. And in fact, our original goal in conducting the study was to enroll about 35,000 participants. So we far overshot that. This chart is just showing you kind of a breakdown of cases. So bipolar, schizophrenia controls, and the total number of samples across different sites. We have one site in Uganda, primarily. We have a lead site in Uganda, a lead site in Ethiopia, 2 sites in Kenya, and 1 site in South Africa that's like kind of leading the study. But you can also see the sort of smaller recruitment sites scattered across these countries. So some new biology that I was kind of alluding that pops out when we do GWAS of psychotic disorders, schizophrenia and bipolar disorders. We're primarily identifying associations for schizophrenia. Notably, because schizophrenia is so polygenic, even though it's highly heritable, the first studies of schizophrenia that were done at this scale, so around 11,000 cases, 17,000 controls for schizophrenia in particular, in European ancestry populations only identified a handful of associations. That's also what we're seeing here. So for schizophrenia, for example, we're identifying 2 intronic variants at genome-wide significance. One's in the gene PTPRD, another is in TMEM150C. This PTPRD association is really interesting. It's already been shown in some previous studies to be implicated in bidirectionally inducing pre- and postsynaptic differentiation of neurons. So clearly it has some neurodevelopmental relationships. It also impairs synaptic function and behavior in knockout mice. So if you knock out the gene, you see a clear effect that seems relevant to this disorder. And it's also been associated with neurodevelopmental disorders, particularly things like autism spectrum disorders. This particular variant is kind of interesting also because it's present at about 0.5% frequency in neurogap and basically absent everywhere else. When we've tried to combine our data, so we know that we're only identifying like a handful of associations in neurogap with the sample size we have now, but there are far larger sample sizes that are available, particularly in European ancestry populations and to a lesser extent, East Asian ancestry populations, to try to understand this basis. We're seeing pretty consistent alignment between our loci. We're still grappling with a couple of puzzles. So even though they're very well aligned, we're not seeing a significant increase in new genetic associations that are popping up. And in fact, we're seeing fewer genetic associations if we do the meta-analysis compared to if we look at the European analysis alone. One hypothesis for this is that we're actually removing some of the variants that are correlated but not causal that are popping up as genome-wide significant. So we may have like a fine mapping angle here that may be very fruitful to go after. Some of the really cool things that we can kind of come back to in this part of the world are thinking about the impressive, huge breadth of ethno-linguistic diversity present in these countries. So on average, there are in total, there's about 90 languages that are being spoken by individuals. If you're looking sort of up the family tree, you can see that mom and dad, compared to the participants themselves, are speaking about a similar number. And if you go up to a grandparent generation, you're seeing that these folks are speaking, the grandparents are basically like speaking fewer languages than the participant. We have up to 3 languages that can be endorsed or people say they speak in each of these participants. They also report up to 3 languages for their mom, their dad, and their grandparents. So we have impressive kind of breadth of demographic information that we're intaking as part of this. And we're very interested in trying to understand how genetics, linguistics, ethnicity, and geography are all mapping together. So to that end, we are looking at sort of 3 metrics. We are looking at measures of relationships between genetics, language, and geography, and we're looking at all these different sets of variation in different ways. So for genetic data, we looked at both autosomal and X chromosome variation. We were kind of interested in understanding whether there's sex-biased demography, whether there are differences in transmission of cultural variables, for example, depending on maternal lineage versus paternal lineage. So we looked at the first few principal components, which distills ancestry information into a sort of score for autosomes and the X chromosome. For geographic coordinates, we used 2 sets of coordinates. One was the location of recruitment, which didn't vary a ton. We have like the main recruitment site, but we also use their language data and anthropological databases to map where the center of the languages that they speak originates. So basically, we're giving them an ancestral location based on their languages. Then for linguistic variation, we're comparing individuals based on the language sounds that they speak. So these are using phoneme presence. So these are like which consonants are shared, which vowel sounds are shared across different languages. We're looking at sort of the similarity and differences across populations to also compare these types of relationships. So we can use these types of metrics to then rotate and scale our matrices into a similar plane using a method called Procrustes analysis. So this type of method rotates and scales one set of coordinates to another set of coordinates by minimizing the distance from one to another. This provides some similarity metrics and also visualization strategies to try to understand their overlap. So this gives us an ability to basically compare how similar the relationships are between genetics versus geography, language versus genetics, and language versus geography. So I'm quickly going to summarize that our really fantastic lab member, Mary Johannes, originally from Ethiopia, has done this analysis. And what she has found is that when we're looking at genetics versus geography, we're definitely finding that these language-based coordinates are performing better than their recruitment sites. Makes sense. We're also seeing an interesting kind of increased correlation for the sex chromosomes compared to the autosomes, which is potentially suggesting that depending on where your mom is from or where your dad is from, there may be more or less mobility along that lineage. And what we're generally hypothesizing here is that your mothers tend to be moving around less than fathers effectively, based on this sort of sex-biased correlation. Language and genetics correlate a little less well than genetics versus geography, but interestingly, the highest correlation of anything was our language and geography, which is maybe a little intuitive, but is also super interesting to see in this incredibly ethno-linguistically rich part of the world. Okay, so I'm wrapping that part up and I want to take sort of a hard transition towards dissecting pleiotropy, so the genetic basis of multiple traits altogether. We've been really interested in trying to look at lung diseases for a variety of different reasons. So one of the lung diseases that we're really interested in is chronic obstructive pulmonary disease, or COPD. That's because this is the third leading cause of death globally. We're also interested in asthma. This is the most common chronic disease in children. And then we're interested in lung cancer, which is the leading cause of cancer-related deaths worldwide. We got really interested in these diseases because of the enormous disparities across populations. So there are huge prevalence differences. We conducted a study in the Global Biobank Meta-Analysis Initiative, bringing together biobanks from all over the world. 18 different biobanks participated. We had hundreds of thousands of participants. When we're looking at biobanks and looking at asthma prevalence in particular, we're seeing an order of magnitude difference in prevalence. Some biobanks are reporting a 2 to 3% prevalence, some are reporting 25% prevalence. So clearly, depending on where you are in the world, there's going to be a very different likelihood of somebody having asthma, for example. We also know that it's very fruitful to take a multi-trait approach to understand these disorders and diseases because they're strongly correlated. We know that the genetic basis is strongly correlated, but we also know from an environmental perspective that while there's shared genetic risk factors, non-genetic risk factors can be somewhat unique to these. So smoking is an enormous risk factor, of course, for lung cancer and COPD, not so much for childhood onset asthma. So one of the first things that we did was combined genetic association results for asthma, COPD, and lung cancer, but also clear known risk factors like smoking, cigarettes per day, and spirometry measures. We're interested in extending this type of work in the future to also incorporate immune cell counts, which also are known to have a clear impact on the etiology here. So there's eosinophilic asthma versus neutrophilic asthma, for example, and they respond very differently to different types of medication, suggesting that it's really important to understand those biomarkers to really understand the heterogeneity and subtypes of these diseases. One thing just to note is these are all very highly correlated. You're not seeing a whole lot of white on the bottom part of this matrix. So you see a lot of things that are positively correlated, asthma, COPD, lung cancer, smoking, and cigarettes per day, for example. And then spirometry measures how quickly you can blow out air into a tube or how deep of volume you can intake into your lungs. You can see that those are oppositely correlated with these sort of negative risk factors. So we tried to develop a new sort of polygenic score approach as part of this big project. We had sort of two sets of types of analyses we did. So the first set is conducting multi-trait GWAS analysis, leveraging the genetic correlation across these traits to improve power to discover genetic variants are associated with each of these traits. So we can leverage the genetic correlation, for example, between asthma, COPD, and spirometry to increase power to detect genetic variants that are associated with asthma specifically, because they're highly genetically correlated. The next part of this analysis was conducting cross-population or cross-ancestry analysis. So leveraging data across multiple different ancestry groups to improve accuracy for each ancestry group. And then we combined all of this together using machine learning approaches like ElasticNet and Lasso. So the first thing I want to show you is what happens when we look at across these different risk factors. Which genetic variants are we finding that are specific to a disease, and which ones are we finding that are shared? So on the left, I'm showing you sort of what's specific to COPD, what's specific to cigarettes per day, and then sort of along, if we're finding horizontal or vertical lines, that's going to indicate what's specific to each. So by and large, you can see that genetic variants tend to be shared across both, but you're finding a bunch of genetic variants that are also specific to COPD. COPD is largely a disease of smokers. About 75% of those who are diagnosed are smokers, but there are smoking-related risk factors, and there's also non-smoking-related risk factors. So a quarter of COPD cases are also non-smokers, and clearly they must have a genetic predisposition as well, suggesting there may be genetic risk factors that are unique to COPD that don't act through your likelihood of smoking more. And so there we're seeing some very known classical canonical risk factors like CFTR, the gene that encodes basically the cystic fibrosis gene. We're also seeing SERPINA1. This is basically a phenocopy of emphysema, a genetic phenocopy of emphysema. So people who have alpha-1 antitrypsin deficiency through genetic variants in the SERPINA1 gene have emphysema despite never having smoked necessarily. We're seeing similar things if we look at lung cancer. So by and large, lung cancer is increasing your predisposition, or smoking is increasing your predisposition towards lung cancer. But we can also see some very interesting and unique risk factors towards lung cancer. So for example, we can see the BRCA2 gene is popping up here, as well as variants in TERT, the telomerase gene that's a known cancer-related gene. These have potential implications for how therapeutics are going to work if people have genetic variants in these genes. Some therapies may work better than others based on tumor pathogenesis. Okay, I think for the sake of time, I'm going to skip this part and just launch into the last little bit. One of the areas that we're most excited about that's really exploding right now in the field is in proteomics. So we're interested in this because proteomics has a far more proximal state to disease compared to a genetic risk factor. We can use genetic risk factors to predict disease earlier in life than you might with other other molecular biomarkers, but proteins sit much more closely to disease state, and they're much more dynamic. So that's really exciting. People have done studies using proteomic data from the UK Biobank to try to predict a wide variety of different traits. I've been talking about respiratory traits, and so I just wanted to mention we're able to predict these diseases very accurately. So we're able to predict death quite accurately, we're able to predict lung cancer quite accurately, and COPD quite accurately as well. The breakdown in these models, so why are some of them blue, why are some of them green? The green model is just a basic model where we have age and sex and we're predicting death. So that alone is doing a pretty good job. If we add on a protein score, we're predicting death far more accurately. If we have an expanded model, we're doing a little bit better at baseline. The protein model doesn't really change things much. But one thing that we saw that we thought was really interesting when we were looking at this other study is that these expanded models that include things like BMI, alcohol use, social deprivation, educational attainment, and importantly for these diseases, smoking, when you look at the baseline model compared to this expanded model, there's basically no difference between the protein score and this expanded model. So that suggests to us that environmental risk factors are very, very well captured by protein scores and proteomics in general. So we launched into a deep study to try to dissect how much of the proteome's ability to predict disease is genetic, which has been largely attributed to a lot of these types of outcomes, versus how much is environmental. So I'm going to spare you some of the methodological details, but I'm just going to tell you that from our analysis, we find that only about 8% of incident disease that we're associating, so predicting disease into the future, given your proteomics today, has Mendelian randomization evidence suggesting that this is the causal protein driving this disease. And it's uniquely specific depending on which disease you're looking at. So most things are non-causal for disease, but are quite correlated with it nonetheless, suggesting there's some environmental perturbagen that's impacting your protein scores and your proteomics overall. So one thing that we were really interested in trying to dissect is we trained a protein score using smoking status And we're very interested in understanding how accurately we're able to predict an environmental exposure with clear known impact on these diseases like lung cancer and COPD. So in the UK Biobank data, we split our data in half. We use that half to train, the first half to train our model using lasso penalized regression. And then we use the other half to ask how accurately are we able to predict smoking based on never smoking status versus current smoking status. We predicted smoking status incredibly well. We had an AUC of like 0.96, which is some value that I'm never going to see again in any of our prediction models. This is unbelievably accurate. We also saw that a huge portion of the proteome, so 66% of disease-associated proteins, were significantly associated with smoking. So smoking is basically a battering ram to your proteome. We've repeated this type of analysis with alcohol use and seen that even more of your proteins are actually impacted by how much you drink. So this is very interesting and has a lot of potential implications for biomarkers like proteins. I'm just going to kind of skip through this for the sake of time and just mention that these biomarkers are also really dynamic. So for example, on the left part of here, we're seeing stratification in current smokers compared to never smokers, and then a little bit of a shift between never smokers versus previous smokers. But within previous smokers, we're also seeing stratification in time since people quit smoking. So for example, we're seeing a difference between 0 to 2 years, 2 to 4 years, 4 to 20 years, and over 20 years ago, people stopped smoking. And then we're also able to stratify current pack years. So this is basically like the lifetime accumulation of how much somebody has smoked over their life course. The fact that we're seeing this is really interesting and suggests how dynamic this is and how useful this is as a potential biomarker, even if we don't have environmental exposures measured in one biobank or one population. Using data that's trained incredibly accurately in another cohort, we could potentially understand your environmental exposures through these types of measures. So this gives us a tool to map from one biobank or one study to another study and predict and impute missing values that are really impactful and important for human health. So maybe this is one of the precision medicine tools that we need since we seem to be very bad at aggregating in a harmonized way environmental data that's really important for human health. Okay, I'm going to end there and just stop with my takeaways and say clearly diverse genetic studies are the best for everybody. Pleiotropy dissection, looking at multiple traits together and trying to understand their genetic variants, are a very powerful but underutilized path forward to understanding specific molecular mechanisms that are impacting different diseases. Genetics provides a uniquely powerful tool for understanding heritable disease and putting things in a causal inference framework. So not just associating correlations, but also understanding and following all the way through which variants are most likely causal versus not. Genetic technologies, I think, are very helpful in potentially improving our clinical models. So in cardiovascular disease, for example, we have a pooled cohort equation that considers like your obesity likelihood, your smoking risk factors, whether you have hypertension, what your blood pressures are, your LDL cholesterol. You could just consider a polygenic score as one more clinical risk factor that you could pile on top of those and build an aggregate clinical score that's going to perform better than what we currently have today. However, I think our genetic study biases are likely to increase disparities given that they're less accurate in non-European ancestry populations. And then I also think molecular biomarkers are really exciting and they're coming for precision medicine, but our ability to delineate molecular mechanisms from confounding are poorly understood right now, and we need to do deeper studies on those. So I'm going to stop there. Thank you all as my hosts for being such wonderful, gracious hosts. Thank my lab and other folks for doing so much of this amazing work. Thank you.
¿Preguntas?
Thank you very much for the talk. You mentioned that you could use a multi-trait GWAS to incorporate like asthma, lung cancer, because those traits are highly correlated. I understand that by doing so, you are like abstracting the traits to a more general notion of like lung diseases. But I wonder if it wouldn't be better to do like the opposite, because for example, lung cancer can have a lot of subtypes, and those subtypes might be related to specific cell types or subcell types. So if you have more accurate or detailed data, wouldn't you be able to be to identify to identify better or more specific genetic markers for those types that might allow to more specific possible genetic targets, for example?
Yeah, this is a great question that we get all the time, especially from folks who are more clinically minded. They are very interested in the details of their specific phenotype and know about its whole disease course. They know about the biomarkers that are related, the diagnostic criteria that may vary between different subtypes. They also know the progression and disease course and which patients are going to respond to different diseases. I think we need, for this particular discovery piece, one thing we need to keep in mind is that we're really early on the discovery trajectory for a lot of diseases. We're underpowered to identify new genetic associations. So we've looked at what happens if you lump versus split. And effectively, what works best is grouping diagnostic criteria together, analyzing a large phenotype together, and not paying quite as much attention to the diagnostic criteria for discovery purposes. That's not true for follow-up at all. But for discovery purposes, the largest sample size is effectively the most important for most disease areas right now. And then you can take those genetic association results that have been identified and follow them through in more carefully clinically curated disease cohorts to try to understand, is a polygenic score really working well in lung cancer patients that cluster with tumor oncogenesis sort of pathways or those sorts of things compared to those lung cancer patients who are smoking a lot but don't seem to be having a predisposition in known tumor oncogenesis pathways. So yeah.
I have two questions. One is, in the search of these genetic variants, is there a measurement of homozygosity and gene penetrance?
Homozygosity, no, although we do measure degrees of relatedness. So in our models, we basically use, we mostly use linear mixed models that incorporate things like your ancestry and your genetic relatedness. If you have a lot of homozygosity, you tend to have less genetic variation overall. And so you're going to look a lot more genetically similar to other people who also have a lot of homozygosity within that population. So I guess no is not quite the right answer, but not really is a better answer. We haven't looked specifically at homozygosity in these large biobanks because it's a little bit more complicated to do. We're kind of going after the simplest thing to aggregate the largest data set. And then your second question was about incomplete penetrance. So there, again, not really. Because we're looking at complex traits, everything is a little bit incomplete. If we were looking at rare disease, then it would be very important to try to think through carefully incomplete penetrance. And in fact, people are very interested in that. For our complex trait analyses, things like type 2 diabetes or cardiovascular disease or asthma or something like that, we're simply getting a measure for every genetic variant. This is the increased risk if you have this genetic variant. These are very small effects. They're just scattered all over the genome. So we're going to have an odds ratio of like 1.1 or 1.2. That's very weak in any specific genetic variant. Is going to be very hard to study its effects because they're so small. So that's why we need to take on some of these multi-trait and clustering of genetic variants to have statistical power to start to tease apart and understand the molecular biology underlying how a set of genetic variants are working together to increase risk. For example, we're looking in thyroid cancer and we're seeing thyroid gland development pathways. We're also seeing tumor oncogenesis kind of pathways that are clustering distinctly, and understanding how those are acting is really important, but doesn't get us at all towards the question of incomplete penetrance.
Cuando si uno toma la variación, the genetic variation, sorry, the genetic variation as an indicator for treatments of the particular disease, Is it considered if the gene is actually acting during embryogenesis? Because if the is not really active before birth, there is no target for it.
Great question. No, is the short answer. For thinking about how genetics should be impacting thoughts around therapeutic development, everything that we're looking at is complex disease risk. So again, most of the diseases we're looking at are adult onset or later onset diseases. Some of them are slightly earlier onset, like asthma. But in those circumstances, the therapeutic development that has exploded has been around which of the genetic variants are perturbing specific molecular pathways. Like in asthma, for example, there have been antibodies to IL-6 and other interleukin genes that have been biologics that have been remarkably successful in the last few years, but they're also incredibly expensive. None of these are related to whether the lungs didn't fully develop, for example, prior to birth, like whether someone was premature when they were born and whether that impacted their lung development. But they are impacting mostly inflammatory pathways and those sorts of things. So most of the therapeutics, I suppose, that we're looking at right now are kind of like later onset.
Hi, thank you. Great talk.
Thank you.
I have some questions, two questions. The human genome, as other mammal genomes, is full of splice forms. And I see that all your studies are centering more in the proteome. Are you able to be accurate about what splice form is being affected? And the second question is whether your methods, are you able to say how much is affecting the regulatory genome? And are you going towards, because many of these genomes are regulated by many different kinds of regulatory elements.
Really fantastic question. So the first part, looking at splice variants or specific types of coding variants that are and what their functions may be. When we're doing whole genome or whole exome sequencing, yes, we definitely are looking at different splice forms and trying to understand whether this is impacting the canonical isoform or whether it's impacting non-canonical transcripts that are expressed in specific tissues. Now, with our proteome data right now that is available from the UK Biobank to everybody, those are using new technologies that are cheaper for doing like large scale analyses, but these are adaptamer-based and antibody-based technologies. So they're mostly grabbing just the canonical and most widely expressed isoform. They're not pulling down like a specific isoform at the moment. So that's definitely like a big limitation. One of your other comments was about like the regulatory component, how much of our genetic variants that we're identifying are regulatory. In GWAS, it pretty closely reflects how much of the genome in general is non-coding. So, you know, like 1% of the genome is coding, about 2% of GWAS variants are coding. So there's like a slight enrichment towards coding variants, but it's very slight. And then yeah, we're looking at non-coding regions mostly through like QTL type analyses, so protein QTLs, eQTLs, those sorts of things, and trying to map those onto GWAS. It's really limited right now. There's some really nice work from Shamil Sunayev's lab looking at how well we're able to co-localize genetic variants, so understand their eQTL effects and other regulatory effects, and say, you know, basically a genetic variant is impacting expression in this way, which is impacting proteins in this way, which is impacting disease all the way through. Less than 10% of the genetic variants that we're trying to co-localize actually do co-localize, giving us quite a big problem with head scratching about what the heck all of these GWAS variants are doing.
I will read a couple of questions from YouTube. Dr. Esperanza Martinez asked whether air pollution, whether you have some data about air pollution and prediction of diseases like Alzheimer's disease.
Oh, that's an interesting question. We have been thinking about air pollution in the context of asthma and other lung diseases. So we've been working on developing protein scores, for example, for lung disease, including smoking, alcohol use, social deprivation. We've been planning to put pollution on that list, since obviously it has a huge impact. But we haven't been thinking about this in age-related diseases. I do have a colleague who's very, very interested in aging and dementia and is using proteomics data. So I should ask him if he's working on that. I have no idea.
So the other question is from Yuridia, and she asks, how can precision medicine help a patient when there is a precision in risk prediction?
I'm sorry, can you repeat that?
I don't know. So how can precision medicine help a patient when there is a precision in risk prediction? There is not, maybe.
There's imprecision, maybe.
Yes.
Yeah. So one thing I like to think about in this sort of consideration of molecular biomarkers is, we can do an okay job predicting your risk of cardiovascular disease, your risk of breast cancer, a risk of a wide variety of different public health priorities that impact a huge portion of the population. Right now, we're using kind of low-hanging fruit to try to get what your risk is. So like I mentioned, for cardiovascular disease, we'll take your blood panel measures, which are relatively inexpensive to do. We'll take your blood pressure. We'll ask if you smoke. We'll take your BMI, those sorts of things. Genetic data is not that expensive to obtain. So I mentioned like BGE is about $100 per sample, but chip-based data is even cheaper than that. Compared to treatments of those diseases, they're much cheaper. But if we're trying to think about proteomics, which is a lot more dynamic, but also much more predictive, those are very expensive. Like they're on the order of hundreds of dollars per participant or per individual. So one thing that we've been kind of thinking about is whether the predisposition towards risk should be a screener also for who gets additional molecular biomarker technologies run, such that we're then better able to determine who gets the most expensive treatments. This is, I think, especially important in cancer, where the treatments are unbelievably expensive and a huge public health burden. But some of the molecular biomarkers are a tiny, tiny, tiny fraction the cost of what the treatment is. So imprecision across populations will have an impact, particularly for genetics because of the ancestry issue I mentioned. It may also have an impact for some of these molecular biomarkers. The largest studies that have been done using proteomics to date have been in the UK Biobank by far. They're coming in all of us. There's also common in TopMed, which has ancestral diversity as a priority. But we have very limited idea about how some of these biomarkers are going to generalize. We know that genetics generalizes very poorly. Proteomics may generalize a little bit better, and some of this may be impacted by the precision of our data setup, like, for example, UK Biobank having higher heritabilities for a variety of kind of natural variation in the population than elsewhere.
I will read the last question.
Okay.
This is from Amara, and she says, great talk. And the question is, how do you think that including and analyzing mitochondrial genome can affect the accuracy of risk predictors of various diseases?
It's a good question. The mitochondrial genome is 17 kb. It's a really, really small fraction of the overall genome. So it's actually relatively straightforward to analyze. There is some fantastic work from Vamsi Mutha, Ben Neal, and Rahul Gupta is the first author that I would encourage looking into. They've done sort of an analysis of mitochondrial DNA and how it's impacting risk of a wide variety of diseases. There's been a standing hypothesis in the field that people have, lower mitochondrial function in a wide variety of diseases, and this is what's driving risk towards depression, or what's driving risk towards fatigue, or a wide variety of different things. They've systematically undermined every single, like they've kind of been able to say these hypotheses don't seem to be playing out for a whole bunch of different diseases. I think their reviewers wrote something like their paper was effectively like the de facto guide to mitochondrial analysis in the field, and that's come out relatively recently. I think that was in the last couple of years. So if you're interested in mitochondrial genetics, I think Rahul Gupta has written one of the most fascinating, fantastic papers, and I think it was published in Nature, 2024 maybe.
Okay, thank you very much, Alicia.
Thank you.